Hello, good afternoon, ladies and gentlemen. My name is Kingsley C Anukam, and I'm happy to make this presentation. Why do humans smell? The auxiliary microbiome compositions of men and women in Nigeria using deodorants. Now, the skin microbiome or the skin microbiota is composed of bacteria, fungi, viruses, and mites. And these microbiota dialogue or communicate with resident dendritic cells, which kind of are resident, resulting from the complement activation. And the skin microbiota is shaped by numerous factors, such as the extrusive factor, which will come up as a result of occupation, hygiene, use of drugs and cosmetics, even including the geographic location and seasonal or climate seasonality. That could mean during dry season or during wet season, rainy season, the skin microbiota are all different. Then the intrinsic factors, and it could be the genetics, the sex, and then size of the body. And then some studies have shown that the skin microbiota have a role to play in developing certain dermatological disorders such as acne, atopic dermatitis, psoriasis, and seborrheic dermatitis. Now, one could ask, does age really matter in the skin microbiota composition? And result studies show that children born through the cesarean section and those born through the vaginal birth have a different skin microbiota composition. Even infants, as those who are known as toddlers, and children that are growing up, and then adolescents, even men and women, all have different skin microbiome compositions. And even those who are considered as aged or seniors, they probably have a different skin microbiome composition. Now, one may ask, do Black Africans harbor distinct skin microbiome? Some early reports have suggested that yes, race play a role in the composition of skin microbiota, but mainly skin microbiome or skin microbiome is really affected by the site of the body. Now, in some studies carried out between Cameroonians and the Japanese, and research studies show that the forehead, forearm, and the back of Cameroonian and the Japanese, comparatively, they have a relative abundance of the general found there. Especially the Japanese harbor more relative abundance of Cutibacterium, while the Cameroonians have more relative abundance of Staphylococcus aureus. So, which means a race actually has a different skin compositions, depending on where the person comes from. Now, one may ask, why do humans smell? The human body is produced by bacterial transformation of the odorless precursors molecules secreted into the surface of the skin by the apocrine glands, and which open into the hair follicles, typically occur in high density at specific body sites, the axilla, the underarm, the nipple, genitalia. Now, research studies published by the Nature Research and Scientific Reports was able to delineate the molecular basis of the alcohol production in human body odor. And then in human body, and there's a link between the axillary apocrine glands, the underarm bacteria, and the body odor, which was first recognized over 50 years ago. Now, early culture-based found that the axillary microbiota to be dominated by the Staphylococcus, Corynebacterium, Propionibacteria, and with Corynebacterium correlating with malodor intensity. Now, it's interesting to note that about 75% women have more 75% more apocrine glands in the armpit than men, but male apocrine glands are larger and may be more active in order to supply nutrients and for bacterial growth. So it may be interesting to know that both women and men, they have the apocrine glands, but male partners have larger apocrine glands. So which may translate that men really do smell more than females. So if you look at these apocrine glands here, the male glands are larger, why the female ones are smaller. So this might contribute to the differences found in the odor being produced by these two gender. Right? Now, the apocrine glands secrete lipids, proteins, and long-chain fatty acids, in contrast to the acrine glands that are found in high density all over the body, and which opens up directly for the thermoregulation, as it contains many metabolic waste containing salts and water and other electrolytes. Now, in the other arm of the axilla, malodor arises due to the biotransformation of dipeptide conjugated to alcohols. So this biotransformation of these molecules, when it's being released, that what produces body odor. Now, the cysteine glycine 3-methyl 3-sulfonyl hexane 1-ol, they are actively transported by peptide tripeptide transporter by certain bacteria, especially the Staphylococcus hominis, Staphylococcus multicus, and lugdunensis, have all been associated in the production of thioalcohols. To expatiate more, the malodor precursor is being transported by tripeptide across the cell membrane of the bacteria. Now there's a cleavage of this glycine molecule by this diaminopeptidase enzyme, releasing the cysteine 3-methyl 3-cysteine hexanediol. Now another enzymatic reaction being part of the cleavage releases the thioalcohol. This thioalcohol is kind of transported outside the bacterial cells. So when it's being transported outside the bacterial cells, that's when the body odor starts occurring. Just as explained, so inside the bacterial cell, this precursor terminal glycine is cleaved by that peptidase, and then releases the cysteine 3-methyl 3-sulfonyl hexane 1-ol, which is exported outside. So the human body contains numerous chemicals, but the most pungent, recognizable ones are just the thioalcohols. Then the hygiene habits, such as shaving the axilla, the use of deodorants, antiperspirants, may alter the odor profile by changing the volumes and the microbiota profile and its metabolic activity. Some cosmetic creams contain nutrients such as the glycerin, amino acids, and hydrolyzed collagens for resident microbiota. So most of the cosmetics some people use contains all these components that help the bacterial organisms to proliferate and grow. Now, we did a study at St. Andrews University whereby we looked at the composition of the axillary skin microbiota of men and women, asked it, and then we looked at the effects of those who are using deodorants and antiperspirants on the bacterial communities. So in study, we've discovered that 26 phyla we identified in males, with actinobacteria being the most abundant, followed by Firmicutes and the Proteobacteria and Bacteroidetes. Then 25 phyla were equally identified in females, and actinobacteria was the most abundant phyla found there, followed by Firmicutes, Proteobacteria, and Bacteroidetes. So in this graph, you will see that actinobacteria, Firmicutes, and Proteobacteria, followed by Bacteroidetes. These are the most 4 abundant phyla found between men and women in the axillary skin. Now, a total of 747 genera were identified, out of which 74% for all are common to both males and females, and out of that 21% were exclusively found in males, while that 3.8% were exclusively found in females. Corynebacterium was the most relatively abundant genera in both male and female subjects, followed by Staphylococcus aureus, Anaerococcus, and Propionibacterium in that order. Now, in this graph too, I will show you the relative abundance of the taxonomic genera in both in male and female subjects, and that shows that Corynebacterium, Staphylococcus, Anaerococcus were the most abundant genera found in these two categories of subjects. Now, when we try to find out the relative abundance or at the species level, we found that the Corynebacterium appendicis was the most relative abundant species found in the axilla of male subjects. And at the same time, the female subject, so what was found there to be the relative abundance of the Corynebacterium was the tuberculous stericum, was the most relative abundant Corynebacterium species found in female subjects. Now, there was really a significant difference between males that reported regular use of antiperspirants and deodorants and those who reported non-use of deodorants or antiperspirants. In the relative abundance of the Corynebacterium general, so 68%, and then the rest was 42%. Male subjects that reported regular use of antiperspirants or deodorants had more relative abundance of Staphylococcus species than the male subject that reported non-use of antiperspirants. So when we looked at the Staphylococcus species relative abundance, those species that are being incriminated in causing odor, we found out that Staphylococcus hemolyticus was the most relatively abundant found in subjects that use regular deodorant antiperspirants. So that will tell you that the Staphylococcus producing odor producing bacteria are found in those who use deodorants and antiperspirants, which means the more you use those antiperspirants or deodorants, the more you produce odor producing Staphylococcus species, especially the hemolyticus and hominis and the lugdunensis. Now, our study report revealed that lactobacilli in the axilla was found in 82% of females and then over 81% of male subjects, although it was found in a low relative abundance, which suggests that Lactobacillus tarsa might be considered as part of the normal axillary bacterial community. Now, what we do with the lactobacilli we found in the axilla, we wanted to see whether the lactobacillus, if we use the lactobacillus to compound a cosmetic product, a topical product, what would be the effect? So we used Lactobacillus pentosus KCA1, which was isolated from the head vaginal premenopausal women, and the study had been published, I think, in 2013 in PLOS ONE. So, and then the genome sequence of the pentosus KCA1 has already been determined, and all the composition or genomic capability of this organism have been known. And in another follow-up study, the KCA1 was used to produce a topical cream, and one of my colleagues in the Netherlands and their research lab used the pentosus KCA1 to produce a topical cream, which was kind of very useful in treating skin acne. It had a very promising result on that. And then in the result they published in one of the Nature papers, Lactobacillus pentosus KCA1 was combined with rhamnosus GG and lactarum in what they produced, and that was able to take care of those subjects that had acne. And then after a couple of days, they examined the skin microbiome again of the acne subject and found that there was really clearance. So the pentosus KCA1, rhamnosus GG, and lactarum was able to take care of those who have skin acne. Now, what we did with what we produced, with what we compounded with our own with our own KCA1, we looked at the relative abundance of the male and female subjects even before they applied the cream, and then we found out that the Staphylococcus aureus or Staphylococcus species were more in abundance, and then even the Corynebacterium genera too, and then the Lactobacillus even before the application of the very small. But then after 14 days usage of the topical cream, which we made from KCA1. After 14 days, we found out that the relative abundance of the Staphylococcus general and the species were really significantly reduced, while in comparison, the Lactobacillus species were higher, and most of the subjects, reported a very significant control or other control from the axilla. Now, the paper has already been published in a journal of cosmetic dermatology, and we titled the publication as "Topical Cream Containing Live Lactobacilli Decreases Malodor Producing Bacteria," and then it equally downregulates the genes encoding PL-dependent enzyme in the axillary of the skin microbiome of adult Nigerians. Now, there are over 30 top words that are used, in claims for cosmetics marketed as probiotics around the world. So people need to be very careful to know all these creams that purportedly claiming that they have probiotics in them, especially the cosmetic, should be careful, because most of them have not been done, as the clinical trials have not been done. There's no proper research backing up all their claims. So you have to be on the lookout to make sure that the cosmetic cream has claimed to contain probiotics. You have to look for the particular species and the strain that was used in compounding that, and then you have to look for search for the literature to see whether clinical trials have been done, and then whether the organism present in that cream, have all the probiotic characteristics, and whether the genome of the bacteria have been sequenced or even deposited in the NCBI, that's National most of the DNA or gene data bank. You have to make sure that you have access to where you can look after those organisms that are present in the cream. Okay, so these are the people who are working in my multi-biome biotherapeutical research at Nantachuku University, especially Professor Nicos. And this Victoria Omore was the master student that did the compounding using KCA1 as main ingredient in the topical cream. And Professor Jose Kechebolo and Samadina were part of a multi-biome therapeutic research group. And if you have any question, please feel free to ask. Thank you.